We'll talk about the direction of flow. And then the regulation of flow to capillaries, basically how that regulation happens. Regulation of flow. All right, so with arterioles, the bands of smooth muscle here are uniform distributions of smooth muscle, right? So uniform bands of smooth muscle. With meta-arterioles, they're also bands, but they're not uniform. So simply bands of smooth muscle. And then same thing with precapillary sphincters. These are also bands that are interrupted, distributed, and not uniform, right? But they're all in different strategic locations. They're more kind of global within the arteriole, and then strips or bands within the other two structures. Now, direction of flow that they control is from arterioles, they control the direction flowing towards the capillary bed. Right? So constriction of that will close off flow to the capillary bed. Relaxation will increase flow to the capillary bed. And then for the meta-arterioles, it's bypassing the capillaries towards venules. So here we're bypassing the capillary bed. And not capillary bed, but more specific capillaries, right, within a bed. So bypassing capillaries towards venules. Arterioles are going to shunt towards specific capillary beds, larger complex networks of capillaries, but that within a capillary bed, meta-arterioles bypass those specific capillaries and goes towards venules. And then pre-capillary sphincters, similar. It's going to be flow toward the capillaries. And again, these are within a capillary bed. And then if you look at their regulation of flow, what happens when they contract and relax? That's kind of the important point we want to make here. So contraction causes decreased flow here, whereas relaxation causes increased flow. However, at meta-arterioles, because this is bypassing or shunting, we're going to see the opposite. When we contract the meta-arteriole, we actually increase the flow. And then when we relax, that decreases flow, right? Because remember, it's bypassing or shunting. And then the same is true for precapillary sphincters and arterioles because they are towards the capillaries, or in this case, capillary beds. This will have contraction, right? Or contract allows for decrease in flow. And then when we relax, that increases flow. Okay. So the one exception here is the meta-arterioles because they are shunting or bypassing. So when that contracts, it increases flow, and then when it relaxes, it allows flow to move into the meta-arteriole, bypassing the capillaries and going straight towards the venule. Okay. All right, any questions on what we've said so far? Any questions? Okay. All right. Now, what we're going to move into is the third part of this topic, which is the movement of materials across capillary walls. And I just want to give you a heads up. This is a very complex topic. We are going to be talking about forces that move fluid in both directions across the capillary wall. And we're going to talk about some numbers and equations and the Starling's forces. So I just want to give an overview of kind of the depth of the topic before we get this introduction going. So we'll introduce the main forces today, and then when we meet on Monday, we'll add the numbers and start, reiterating, going into the depth of this. So when we think about movement across the capillary wall, it's an exchange happening, so it's movement in both directions, and the most common mechanism for this is diffusion. So because of that, we're going to think about lipophilic substances that can go right across the membrane, which is simple diffusion, right? It can move right through that membrane if it's low in terms of if it's small in terms of its size, if it's lipophilic, and if it's uncharged, right? So when you think about the substances that are lipophobic or larger or charged, they are going to require a channel for some type of facilitated diffusion. So these are the most common mechanisms that we see for movement across capillary walls. We also have transcytosis. This is where we have proteins, especially packaged in vesicles, and we have endocytosis and exocytosis, which moves these packaged proteins across the cell. And then in the brain, we have even more specific type of transport called cell-mediated transport because of the presence of the blood-brain barrier. So the blood-brain barrier is a very specific, very unique location for transport. And so we do need a more specific mediated transport in that example. So these are some of the examples of how we move things across in different capillary beds, but it does require us to talk about the forces that move on the capillary wall. Now, when you think about bulk flow of fluid across the capillary wall, it's based on gradients, right? We've described some of these gradients before. We want to have high pressure in one area, low pressure in another area. And so that creates a passive gradient where movement can happen naturally in terms of that flow. Protein-free plasma is what's really going to move across capillaries, right? The most abundant being continuous, and then the pores in those continuous capillaries not allowing proteins to pass through. So the plasma accepting those larger proteins is what is moving through. Filtration is movement out of the capillary into the interstitial space. So thinking back again to AMP1 here with our different fluid compartments, right? We have intracellular fluid inside the cell. We have extracellular fluid outside the cell. And within that extracellular, we have interstitial, which is fluid around the tissue. And then we have plasma, which is fluid in the capillaries and vessels. So that movement taking place within the extracellular fluid going from the capillary plasma into the interstitial space, interstitial fluid, is the direction of filtration. And then absorption is in the opposite direction. Fluid that's moving from the ISF back into the capillary will be happening as well. So there's movement in both directions. And so here we want to think about what are the forces acting for filtration, what are the forces acting for absorption, and then we're going to want to add those forces together and subtract the stronger force from the weaker force. That is going to tell us the overall direction of movement of plasma, whether or not it's going into the interstitial fluid or whether or not it's overall being absorbed back into the capillary. All right. And so the whole purpose of this type of movement is to have equilibrium, to redistribute the extracellular fluid and have some balance between these two ECF compartments. Right. So reminding ourselves here, this is a continuous capillary. These are the pores in between the endothelial cells. This is the lumen. These are the cells stacked against each other. And then looking at a cross section of this, we can see the different types of exchange, different types of movement that's happening. So here's the plasma. Here's the interstitial fluid or tissue fluid, both subcompartments of the ECF. And then in terms of the diffusion, What we describe to be simple diffusion, oxygen can move through, CO2 can move through, fatty acids, which are lipophilic, steroid hormones, which are also lipophilic, can move right through the cell and move right through the endothelial membrane. All right. So small, uncharged lipophilic substances can usually diffuse right through. Now, facilitated diffusion is going to be for larger substances, proteins like glucose, as well as charged molecules like ions, sodium, potassium. They're going to require some type of help, right? Either a transporter or an ion channel or some type of facilitated carrier in order to get them through the endothelial membrane. And this is going to be, again, water-soluble or lipophobic substances or large and charged. In terms of proteins, we can also have transcytosis. So endocytosis into the cell, exocytosis on the other end of the cell membrane, and then we can move bulkier, larger molecules right across the endothelial cell as well. And then water, we know by osmosis can move right through the pores, so water can move as well. Now, let's talk about the Starling's forces, the forces that are acting on the capillary wall to move these particles, and then specifically the plasma here in one direction or the other. So this is described as Starling's forces. And there are two types of forces acting in order to give us our overall flow. So we want to look at the two forces individually, and then we'll sum them together and think about the collective direction of the bulk flow. So the hydrostatic force. Hydrostatic force is a push force. It's force based upon the pressure gradient acting across the capillary. So on one end, at the beginning of the capillary, we have a high gradient or high pressure. On the other end of the capillary towards the end, we have a low pressure. So because of that pressure differential, we have a push force, right, high to low, that aims to move fluid out of the capillary and into the tissue, and that is called a hydrostatic force. Okay, now I use the example of out of the capillary into the tissue, but hydrostatic force can be acting in any direction, but its overall direction is pushing. Okay, the osmotic pressure, on the other hand, this is an osmotic force that exerted by the water, and this is exerted by non-permeant solutes. Remember, we talked about this before. The osmotic gradient is a pull force, and it's exerted by the particles that cannot themselves move, or the non-permeating solutes, and that wants to pull water or fluid in order to create an equilibrium in terms of the solute concentration. So an osmotic pressure or osmotic pressure gradient is attempting to pull fluid in order to equalize the pressure gradient or the concentration gradient. And so that is a force in an opposite direction. So only non-permeating or solids that cannot move across the membrane will exert an osmotic pressure, and this is mainly proteins. So mainly proteins existing on one space, because they're so large, they can't cross the membrane. Instead, they pull water to them, which exerts that pulling force on our cell wall, right, on our capillary wall. And so the oncotic pressure is how we describe this here. Oncotic means protein pressure because of the osmotic force of the proteins that cannot move across the capillary wall. So the overall takeaway here, we have two forces. Hydrostatic is a push. Osmotic or oncotic is a pull, and they're both acting on both sides of the cell wall. So in our capillary, we have a push and a pull. In our interstitial fluid space, we have a push and a pull. Right? Okay, so let's think about that, right? Illustrate some of those concepts. So we are looking at Starling's forces. And I'm gonna just describe some symbols here. So P, this large P is representing the hydrostatic force. And this, we said, is a push force. And then we actually use the pi symbol to represent the osmotic force. And remember, we said we're going to equate this to proteins or oncotic, and that is a pull force. Okay. Now let's describe our or our two tissue and fluid spaces. So we have the tissue. This is our tissue up here. In other words, our interstitial fluid compartment. And then we have our, sorry, this is our tissue, okay? Down here is the tissue space. Let's put our capillary in. Here's our capillary. And so this is the tissue up here. This is the tissue space, and this is the actual plasma space. So here's plasma. And then here is interstitial fluid or interstitial fluid, interstitial space. Okay. Now, when you think about the arteriole here, on this end is our arteriole. On this end is our venule, which makes this structure our capillary. And so we're looking at the forces acting on the capillary wall, as well as forces acting from the interstitial fluid space. Okay. And so let's talk about our push forces first. We have two push forces. We have a push force in the interstitial fluid space. So this is our large P, and we're going to put a small IF to represent the location of that force. So a hydrostatic force in the interstitial fluid compartment is our hydrostatic interstitial fluid force. And that is a push from the interstitial into the capillary. In the capillary, right, in the plasma space, we have a hydrostatic force here as well. So this is a large P representing hydrostatic, and then CAP representing the location of that force. And so this is a capillary hydrostatic pressure, and that is also pushing it in the opposite direction. Right. Now, if we think about our osmotic force, this is going to be acting in both directions, but mainly coming from our plasma. So in our plasma, because we have all of these proteins, right, we've got all of these bulky proteins that cannot leave. We call these plasma proteins. These are proteins that help our blood to clot, right? A lot of different functional proteins that exist in our plasma. And so the oncotic or osmotic force from the plasma is always going to be greater, while the osmotic force in the interstitial fluid is almost negligible because there aren't very many proteins, or there shouldn't be, right? Very many proteins in the interstitial fluid space. So it's very negligible in terms of the plasma force. So let's describe those two forces. So we have an osmotic force acting from our capillary. Remember, our symbol for osmotic is our pi symbol, and then from the capillary. And so because this is osmotic, now it's a pull, it's pulling from the interstitial fluid space back into the plasma. Similarly, we have an osmotic force in the interstitial fluid space, and that is likewise pulling from the plasma in the opposite direction. Okay, so we have both forces acting on the capillary wall in both directions. We have a hydrostatic push from the interstitial fluid into the plasma, and then a capillary hydrostatic in the opposite direction. And we have an osmotic pull from the interstitial fluid into the plasma and an interstitial fluid osmotic in the opposite direction. Okay, so let's describe which of these are going to favor filtration and then which of these are going to favor absorption, right? Remember, filtration is moving of plasma out. Absorption is moving of interstitial fluid into the plasma. So we want to think about which of our forces are going to favor filtration. Right? Filtration, we said, is net movement fluid from capillary to interstitial fluid space. Okay. And when we think about the forces that do that, that's going to be our hydrostatic capillary pressure and our osmotic interstitial fluid pressure. Right? Those are both acting in the direction of filtration. On the other end, absorption is net movement of fluid from the interstitial fluid space to the capillary, right? So opposite direction. And so forces, that would favor absorption are going to be our interstitial fluid hydrostatic pressure and our capillary osmotic pressure. All right. So we're going to think about two different forces that are acting on our capillary, and we'll end it here for today, but we will go on to reiterate this. We'll add some numerical values and actually calculate what the pressure gradient will look like and what direction of flow. We'll talk about some different examples as well. All right, so filtration being our PCAP and our osmotic interstitial fluid, absorption being our interstitial fluid, and our osmotic capillary pressure.